Oh, and then I was close. Recording, remembering to record. See, look, I'm getting a random person trying to come in here too. I don't know who that random person is. Does anybody know if somebody's trying to get in here with a different name? Somebody just wants to learn about microbiology? Okay, bye-bye. All right, there. Well, I'm not gonna let them in, whoever it is. Okay, let's get started with the immune system. We're recording. Okay, I know there's a lot of slides. I think there's 120 something slides. My goal is gonna try to get through some of it today and some of it on Tuesday for the exam on Thursday. So how is the exam gonna work? The exam is gonna have two parts. They're both gonna be on Moodle. The first part will be like multiple choice, fill-in type questions. The second part will be the essay type questions. So the page will pop up, it'll say exam 3. You will go to the multiple choice type questions. You'll have a certain amount of time to finish those. You'll go to the essay type questions. You'll have some time to do those. You can pick which one you do first, but you're gonna have to complete all of it before you can move on to the other one. So if you feel like you want to type for a while, you can do the essay questions first. If you feel like you want to do multiple choice first, you can jump on that, okay? So it'll be open on Thursday, so a week from today, in the same time that we normally do class. Again, if this time can't work for you, I'll send out an email about this too. Please let me know. We can figure out the schedule, but it'll just be easiest. I will also have a Zoom link. So I will be on Zoom that entire time. So if you're going through the questions and you're like, I don't understand what you're trying to ask from this question, you can pop onto Zoom and either chat or just ask me the question directly and I can try to answer it for you. Okay, so let me know. I'll send out another email too to remind you if you have any issues with that time or answering other questions that you can just send me an email and let me know. Okay, so I'm going to try to get through most of the immune system. I know some of you guys in this class are also in immunology, where the tagline for immunology is it's complicated. So all of this that I'm going to go through today and Tuesday for you guys is probably a complete review of what Dr. Thompson and I have gone over in immunology. It's just kind of the Cliff Notes version. So you won't need to know as much detail for this as you would need to know for immunology. And it should be quite a review for you guys. You should have seen that all that stuff before. Okay, so you guys are ahead of the game, at least for these 2 lectures, because you've been doing immunology for the last couple of weeks as well. Okay. All right, so let's talk about the immune system. So we talked last time about how we interact with microbes, and we talked about our normal flora, so all of our bacteria that are on our skin, inside our intestinal tract, that are inside our nose, hanging out in our mouth. So we've got all this normal flora. The other way we interact with microbes is harmful microbes, pathogens, like our lovely friend SARS-CoV-2. My son likes that name for some reason, so he likes to go to preschool and say, did you get SARS-CoV-2? He's funny. Anyway, so we have pathogens. So these are harmful microbes that are not beneficial to us, that they are causing some sort of harm to our body. So we call the invasion of microbes, so the infection, this is called infection, right? When we get a microbe and we have a population of microbes, we call that infection. Okay, so these are intruders to our body. So what do we call them? Microbes that cause infection and disease, we term them pathogens. So these are pathogenic microorganisms. A microorganism that is able to cause disease in any sort of normal, healthy individual. You've got a normal immune system, nothing's going wrong. We call those true pathogens. Those organisms that can cause infection in immune compromised individuals, we call those opportunistic pathogens. So they don't if we encountered them with a healthy immune system, we would not get sick. We call those opportunistic pathogens. Serratia marcescens that we used in the lab, that lovely red bacteria, is an example of an opportunistic pathogen. It doesn't normally cause disease in healthy individuals, but if you, for example, had HIV, it may cause a serious infection because your immune system is compromised, or if you have any other sort of immune disease. Oops. Why am I going backwards? Go forwards. Okay. The severity of the disease, so anywhere from a mild infection all the way up to a systemic infection, we call the severity virulence. So a more virulent strain of an organism cause a more severe disease. Any sort of characteristic that a pathogenic microbe has that contributes to their infection, we call a virulence factor. So I'm going to ask a poll question. So which of the following do you think are a virulence factor? So just by what it does. A toxin made by the bacteria, you can pick more than one. A protein that prevents the immune system from not working, a ribosome, a protein that helps a bacteria enter a host cell. Which one of those do you think would be a virulence factor? Can anybody vote? No? Yes? Okay, go ahead and vote. I'm not seeing any votes. I knew I had trouble with this in immunology, so I don't know if my poll is not working. There we go. I see people. I need to have my poll music. I need to find something on my phone, some Jeopardy music or something. Anybody else want to vote for fun? Give you like 10 more seconds. Okay. All right, most of you guys were able to poll. Yes, so you guys all got it right. Well, most of you got it that a toxin, yep, that probably is a virulence factor. Some protein that helps a bacteria or a microbe evade the immune system, we consider that a virulence factor. And a protein that helps the bacteria enter a host cell probably is a virulence factor. So the ribosome would not be a virulence factor, but all those other ones are probably virulence factors. So anything that potentially helps an organism cause disease, we consider a virulence factor. Okay, go away, Paul. All right, so virulence factor. Okay, so how do microbes get in? This is one of these things when we've been talking about coronavirus that people have been talking a lot about this idea. This is not social distancing. We're supposed to call it physical distancing now. Governor says physical distancing. Okay, physical distancing. This is the reason why we're trying to increase the amount of distance between us and another individual, is because of this idea of microbes entering our body and causing a disease. So how do they get in? There's two types of transmission. One of them we term direct transmission. I'm sick, I'm going to make you sick directly. And then there's indirect, where the microbes are interacting with another surface or another structure before it infects you. Direct transmission, so transmission from me to another person, can be through direct contact. I'll show you my lovely picture of direct contact in the next slide. It can be from droplets. So one of my favorite things for my 2-year-old to do is that he loves to sneeze in my face. He thinks it's real funny. So if my son is sick and he's got some sort of virus in his nasal passages and he sneezes or coughs directly on my face, then his droplets have entered into my mouth or up my nose, and they have directly contaminated with me with anything he has, which is usually the case whenever my son gets sick, is he likes to directly contaminate me. Mother-child is not like that. Mother-child is from in utero. So if you are pregnant, passing it from mom to baby in utero is mother to child. And then biological vectors, these are like mosquitoes or different fly species that are transmitting the microbes via that insect vector. Okay, so that's direct contact. Then indirect, there's one of my favorite terms in microbiology ever is this term called fomites. So what are fomites? So fomites are kind of a generalized term for, imagine when you're talking or coughing and you have anything in your mouth, like you just ate some food and you've got like food particles and you got anything in your mouth and you would cough on something. This is fun. Like here's my mouse right here. Like if I coughed on my mouse, it would have like little droplets from my saliva. It would have anything that was inside my mouth, anything like that. And we term that kind of general stuff that we leave behind as fomites. They're like little particles of food, little particles of saliva. We call them fomites. Indirect also from food and water. I've got some sort of microbe on my hand. I cook some food for you, and then I give you that food and you get sick. That's considered indirect. And then through the air. So if something is aerosolized in the general area, and this is one of the things we worry about with SARS-CoV-2, it's aerosolized, you come into that area and then you're inhaling those aerosol particles in the air. So here's my lovely picture. This is from another textbook, but I keep using it because it's a very good demonstration of these different ways of transmission. So on this left-hand side, we've got the direct methods of transmission, okay? And then indirect on this side right here. So direct, yeah, you kiss somebody, that's some direct contact, right? You're transmitting saliva right from your mouth into another person. That's pretty direct as we can get. Any of you guys have heard that or have gotten mono? That is often called the kissing disease because it's very easily transmitted directly via kissing. I know that in the dorms when I was in school, a lot of people came down with mono. Makes you extremely tired. It's a viral infection, makes you extremely tired, but they often call it the kissing disease. I did have a giant microbe of mono, but I gave it away. I'll have to get another one. It was kind of cute. Had beautiful eyelashes. Anyway, okay, so that's direct contact. Kissing, a sexual contact is considered direct contact. Droplets, aerosols, the difference between aerosols and droplets is the size of the particle. So droplets are considered larger. One of the things you may have heard with SARS-CoV-2, the difference between droplets. So if I cough directly from myself to you, that's considered droplets. They're bigger. They fall quicker too. So it needs to be within, that's why the 6 feet thing, right? If you're farther than 6 feet away from you, if I cough, the droplets should fall down to the earth fairly quickly. So having that distance, I mean, I could be a pretty good cougher, but I don't think I can get 6 feet, pretty good distance away. Where aerosol is more fine particles, and those tend to last in the air a little bit longer because they are not as heavy. So that is where if I coughed, that those little finer particles might remain in the air where the bigger droplets are going to fall fairly quickly. So that's the difference between aerosols and droplets. Droplets are heavier, they're bigger. Aerosols are more fine particles, they stay in the air a little bit longer. You can get aerosols from animals too. Hantavirus. Hantavirus is a fun one. I always tease Dr. Senseneg about hantavirus. Hantavirus is pretty prevalent in our squirrel population. It is a problem with in their droplets, in their droppings, in their poop. If it's aerosolized, it can get in the air and then you can breathe it in and can potentially get hantavirus. And so when he's killing the squirrels, I keep telling him, you're bringing hantavirus inside this building. Keep your squirrels out of here. Yeah, so fomites, again, coughing, touching, any sort of particles that come out of that, we consider those fomites. That's one of the things we're worried about with door handles. I found a wonderful article about copper, saying that copper is a really good antimicrobial and that we should make all of our door handles out of copper because it would cut down the spread of disease from people, coughing in their hands and touching door handles. So maybe after all of this, we can get a bunch of copper door handles and decrease the amount of infection. Okay. And then food, as I mentioned, I have microbes on my hand. I'm preparing some food and I infect you. All right. Okay, so how do microbes get in? Where do they get in? Okay, so those are some direct or indirect. Now, if I coughed on my desk right now, and you touched it with your hand, that's still not good enough for you getting infected. It has to make it inside your body somehow in order to infect you. So this is where the idea of washing your hands comes into effect. And actually, I just have ethanol in my office just for fun, to clean my door handles and everything. So if I coughed on here and then somebody else touched it and then I squirted them with some ethanol, it's better than hand sanitizer, then that would probably it should destroy almost everything on their hands. So that if they then touch their face, they wouldn't bring that through a portal of entry. So let's talk about portals of entry. Your skin is one of your best assets in your immune system. As we get to the immune system, we'll talk more about the skin, but if you got a microbe on your skin, if you didn't have any cuts or any sort of abrasions, most microbes would not be able to get into your body. Your skin is a great layer. It protects you from everything. They have to get into a orifice, or what we consider a portal of entry, before they can cause infection. Okay. So where are our portals of entry? Your skin, if it is any sort of damage. So think of getting a cut on your skin's no longer completely intact, it can get in there. Your GI tract, that's eating anything that's contaminated or drinking anything that's contaminated. Your respiratory tract, your nose, your mouth, urogenital tract, that could be any sort of STD. And then pregnancy and birth, that again is the ability for the mom to give infections to her unborn child. It can either happen in utero, or that's while the baby's in the uterus, or during birth. So a lot of STDs can be transmitted to the baby if you have gonorrhea or chlamydia going through the birth canal. So one of the things that they do test you for a lot of different STDs when you are pregnant. That's one of the set of tests that you get, just so that the doctor knows if you have an STD like gonorrhea or chlamydia, that the baby can be born via C-section and not go through the birth canal, or hopefully you can get some antibiotics to get those treated before the baby is born. Okay, so let's talk about the skin. So the only way for microbes to get into the skin is that the skin has to be damaged in some way. Okay, so it has to have some sort of cut or nick or abrasion, something that has damaged the skin enough that the dermis, we'll talk more about this in the immune system, has been damaged so that the microbes can get underneath the skin. Puncture wounds, including any sort of bites. So we talked about biological vectors a little bit in transmission. So biological vectors, if a mosquito bites you, that's considered a puncture wound, right? Because they've injected, see, this is where I need Dr. Sensenig. I don't even know what that thing is called from the mosquitoes. Anyway, they inject it, try to suck your blood. They can inject microbes into your body when they're biting you. You can also potentially get infections in your hair follicles. If microbes get in there or inside your pores, you can potentially get infections in that area as well. Staph, anyone ever had a staph infection? That's fun. This is an example of a staph infection. Can get in through a small nick in your skin, can infect a hair follicle as well. Streptococcus pyogenes also kind of a skin infection. This is an example of impetigo here. Gangrene, lovely. I forgot to warn you, trigger warning. There's gross pictures today. Gangrene is one of the, it's a Clostridium species. And unfortunately, if you get any sort of burn or wound in your skin, it can potentially get into your skin and cause gangrene. Tetanus. I don't know how many of you guys have ever had a tetanus shot. Hopefully all of you, or you get it once when you're little. And then if you get any sort of puncture wound, anything that's like considered a rusty nail, or like anything that's a rusty object, potentially has Clostridium tetani. That's the bacteria that causes tetanus. It's an anaerobic bacteria. That's why puncture wounds that go deep into your skin, so that it's a potentially anaerobic environment. So if you got a puncture wound, they recommend getting a booster shot of tetanus. I remember, I think I was in high school, I accidentally stepped on a rusty nail and had to get a booster shot of tetanus, and it hurt. It was like a significant needle. Like I remember it being really painful for like a couple of days. Okay, so those are skin infections. The GI tract. So this is food poisoning. I've had food poisoning. Probably most of you may have had food poisoning. Hopefully not from our cafeteria here, but from somewhere else. I got mine from McDonald's cheeseburgers when I was in grad school. I will never eat another McDonald's cheeseburger again. That was once enough for me. I was good. I actually ended up in the ER because I was so sick. Anti-nausea medication is phenomenal. So I recommend if you have really bad food poisoning, probably not now because the ER is a little full, but that you should go get some medical treatment, because those anti-nausea medications can help really with that horrible feeling. Okay, food poisoning. This is consuming food or water that has been infected or contaminated with microbes or microbial toxins. One of the most frequent food poisoning is staph food poisoning, and that is not from being infected with the staph bacteria. It's from a toxin they produce that is left on food, and you consume the toxin, and the toxin makes you violently ill. So we're talking about explosive diarrhea, vomiting, intestinal distress. As we get to specific microbes, we'll talk about the different types of infection. But some examples of these, E. coli tends to be on beef, although E. coli contamination can happen on any food substance that potentially comes in contact with feces. So we had a big outbreak of E. coli on spinach, gosh, probably like 10 years ago, 15 years ago now. And that was from contamination from a dairy farm coming on into that spinach. So the spinach itself didn't have the E. coli on it, didn't grow with E. coli, but it was contaminated, fecally contaminated with the E. coli. Salmonella, in this country, most of our chickens that you buy in the grocery store are contaminated with salmonella. So if you ate a raw chicken, please don't ever do that. Or put some food on a surface that had that raw chicken had been on, it's a potential for getting salmonella food poisoning. Salmonella is one of the easiest food poisonings for you to get. We'll talk about why that is in a little bit. So it is very important for you to cook your chicken. Eggs also have salmonella. Potentially, I know raw cookie dough is very yummy, but there's a potential of getting salmonella from eating raw cookie dough with those raw eggs in there. Most eggs here in the country have salmonella associated with them. Okay. Oh, listeria. If you've ever heard of soft cheeses like queso fresco, any sort of brie or anything like that is potentially contaminated with listeria. Although I remember when I was pregnant with my first son, there was a big listeria outbreak on peaches. So we normally think about it with soft cheeses, hot dogs, also, and any sort of deli meat, often with listeria. Shigella, this is one of the ones that causes dysentery. It's potentially inside water. Cholera is also another water microbes, so you could get cholera from ingesting contaminated water. Respiratory entry, well, our very famous example of this right now is our coronavirus. It has a respiratory portal entry. This is coming through your nose or your mouth, breathing it in through your respiratory tract. Strep throat, meningitis, influenza, chickenpox, the common cold. These are all respiratory routes of infection. Urogenital ports, these are STDs. STDs are normally found in y'all's age group. 18 to like 25-year-olds, 28 years old are kind of the number one for STDs. This is a lovely, from the CDC, kind of a handout about the different STDs. Kind of the number one that's out there are gonorrhea, chlamydia. HPV is human papillomavirus. That's the one that causes cervical, or potentially causes cervical cancer. Herpes, HIV, syphilis is making a comeback. It's kind of the low one on the scale. Gonorrhea is still number one. Go gonorrhea. Gonorrhea and chlamydia are both bacterial infections, so they usually are cleared up pretty easily with antibiotics. There are some antibiotic resistant forms, but mostly antibiotics will get rid of those. Syphilis is also a bacteria. It's a spirochete bacteria, can usually be, you can usually get rid of it with antibiotics. HPV, herpes, and HIV are all viral infections, different types of antivirals. Usually HPV is not treated with an antiviral, but there is a vaccine out there for HPV now. Many of you may have had it. It's not one of the required vaccines. It's a volunteer vaccine, so you don't have to get it. Herpes is usually treated with acyclovir or gangcyclovir. These are DNA analogs because herpes is a DNA virus. But herpes is never completely eliminated from the body. It's just you're able to keep the viral load down. Herpes viruses in general go into latency. So you're able to keep the virus in that kind of latent state. Pregnancy and birth, again, mom can transmit it to baby. Chlamydia is one of the ones we're worried about during the birth canal. Syphilis, HIV, cytomegalovirus, a couple of viruses, herpes can be transmitted from baby, mom to baby. Okay, so those are the infection routes and how we get microbes from one person to the next. So how many microbes does it take for you to get infected from someone else? Will one be enough? If I had one single E. coli on my finger and I licked it, would that E. coli make me sick? Okay. 10? 1,000? Well, it actually varies from microbe to microbe. For some viruses and some bacteria, the dose of how many microbes it takes to make you sick is very low. Some are very, very high. The amount of microbes required to make you sick is considered the infectious dose. So how many exactly microbes that need to be transmitted. Okay, so here's a list of different microbes. This is again from another textbook. And it goes all the way from measles that takes one virus, all the way up to gonorrhea that takes about 100 million cells to make you sick. This is the primary route of infection, so either it's a respiratory virus or something else. So with this list, I'm going to ask my next little poll question. If you're looking at this table, which do you think is the most infectious, the easiest one for you to get? Cholera, measles, plague, or anthrax? Anyone else want to vote? Ten, 9. Okay, I'm gonna close it. Yeah, so most of you guys got it right. It is measles. That would be the easiest spread disease because it only takes one virus particle. So measles is a very contagious disease, whereas you'd have to drink a lot of water to get cholera. So cholera would be kind of on the farther end. Is if we were looking at just how infectious these would be. Somebody was asking about gonorrhea. Yes, you can cure gonorrhea. You can get rid of it. It is a bacterial infection. You can clear it from your bodies. Where herpes you keep for life. Just like if any of you would have gotten chicken pox, like poor me, I got chicken pox when I was a kid. That is herpes zoster. And I have herpes zoster in my body for the rest of my life. Yay. You guys all got the vaccine, so hopefully you will not have to endure herpes zoster for the rest of your life. And that's why the shingles infection is an infection you can get later in life from the herpes zoster virus. So herpes viruses you get to keep forever. Yay. One of the herpes viruses that causes, herpes simplex, that causes cold sores in your mouth. Researchers think that most everybody by the time you're like 30, 35 have that virus, but only very few people show symptoms. And they think it has to do with your genetic background. So not everybody shares or is able to show symptoms. So yes, you can pass herpes zoster. You can pass all herpes viruses from one person to the other. They used to have chicken pox parties when I was a little kid. So I got to go to a chicken pox party and try to infect a bunch of other kids because it was better to get it when you were young. So it's very easy for chicken pox herpes zoster to be passed from one person to the other. Okay, let's move on. Infectious dose. We also have a couple of other terms when we're talking about infection. So the ID50 is a term that people will use for the infectious dose for 50% of the population. So if we're looking at, people from 0 all the way up to, 99 years old, in that sample population, what dose takes it for half of the population to get sick? LD50 is something we use for toxins, like botulism toxin that we inject in our face. LD50 would be for the lethal dose for 50% of the population. So for some microbes, they have multiple portals of entry, and depending on what portal of entry the microbe uses to come into the body, the infectious dose will be a little bit different. Bacillus anthracis is a good example of this that causes anthrax. So Bacillus anthracis, like all bacillus species, produce spores. And if you have a cut in your skin and you get the spores in that cut, it takes only between 10 and 50 endospores to cause the cutaneous, meaning a skin infection of anthrax. Where if you're going to inhale it, it'll take way more spores, so 10,000 to 20,000 endospores. And if you drink it or ingest it in some way, it's even going to take more spores than that. So when they were sending around envelopes, way before you guys were born, full of anthrax, maybe not, to people, this was a very bad bioterrorism microbe to pick. I mean, it was nice that it was in that spore form, so you could send it in a powder form. But it takes so many endospores to make people sick that even from opening the envelopes, you're probably not going to get that many spores in your inhaled it, or hopefully you're not going to lick the whole envelope. It was so few people got sick from it. So it wasn't a really good bioterrorism agent. Terrorists weren't very smart as they were sending those around. Toxins. Toxins, this is the infectious dose for the toxin, botulism toxin. This is how many nanograms per kilogram needed to cause disease in 50% of the individuals. You can see botulism toxin is one of the most toxic that we know about, and great, we inject it in our face. That sounds like a really good idea. Shiga toxin, this is produced by Shigella as well as some other microbes. And then staph enterotoxin, this is the one that causes food poisoning. It is way more grams of the toxin is needed to cause disease. Okay, we talked about portals of entry. We talked about transmission from one person to the next. Okay, so they got in your body. What happens now? How do we get from microbe in your body all the way up to causing disease? Well, through the portal of entry, the microbe gets through some sort of barrier. Then it has to attach to a host cell, the correct host cell. It has to invade that host cell, start replicating, attack whatever tissues it's going to attack, and then from that attack, it will cause whatever disease. The diseases are a little bit different depending on the microbe. The disease can be caused by the bacteria or microbe causing disease and killing the cells, or it could be from your immune system trying to fight that off and the symptoms associated with your immune system. Now, after causing disease, another important thing that the microbe wants to do is it wants to get out and infect other people. Some microbes, the portal of entry and the portal of exit are the same thing. Whereas other microbes, you can think about food poisoning. The portal of entry and the portal of exit are different. It's going to enter through your mouth and it's going to exit through your feces. So those will be different. Although from vomiting, you could also vomit up microbes. So the portal of entry could be the same as the portal of exit. There might be multiple portals of entry, multiple portals of exit. It just depends on the microbe. So can we stop them? Well, that's the goal of our immune system. Depending on the strength of disease and the strength of the immune system, the amount of disease that microbe can cause in your body will vary from person to person. For most people with a healthy immune system, most microbial diseases can be fought off. Some of them can be fought off with time, and so we don't like to wait that amount of time. And we want to have an intervention like antibiotics. Others are very difficult for our body to fight off, so we need to have an intervention, or it's potential that that microbe can cause death. Okay, so what's our intervention? It is our immune system. So our host defenses, the defenses that our body has and other vertebrates have against microbes are called our immune system. And we have two different arms of our immune system. We've got our innate immunity, which is considered non-specific. These fight off any sort of invading microbe. And then we have our specific immunity, and those are specific to a certain type of microbe. So we have a specific immunity for influenza versus E. coli versus salmonella. Okay. This is a nice kind of chart of the immune system that I stole from another textbook. And it goes through your host defenses, and it has a non-specific innate immunity on this left-hand side, and then the acquired or specific immunity here on the right-hand side. And it goes through some different lines of defense, the first and second line of defense. And then it goes through the third line of defense being the acquired immune system. So we're going to start off with this first line of defense here, our innate immunity. These very general barrier defenses against microbes. Ooh, I'm a little fast there. Okay, so innate immunity, non-specific against any sort of invasion. They are present since birth. They don't have to develop like your acquired immune system. Oh my gosh, go forward. There we go. Okay. The first line of defense, like I mentioned before, is blocking microbes from coming into our body. So our skin is a really good defense against invading microbes. It also includes chemical barriers that prevent microbes from entering our body. And we'll talk about some specific ones. Then the second line of defense, we'll go into detail about some of these second line of defense mechanisms. These include cells that do something called phagocytosis. It includes inflammation and fever, and then a chemical called interferon. And we'll talk a little bit about the second line of defense maybe today. Maybe we'll get to them on Tuesday. Okay, so let's talk about our skin. Our skin is fantastic. Thank you, skin. Thank your skin every day. It prevents microbes from getting into our body. If unbroken, it's thick, it's waterproof, and it's impenetrable most of the time. So if it's not broken, our skin is a perfect defense mechanism. We pick up microbes on our hand as long as we wash our hands before we touch a portal of entry. So like if my son loves to pick his nose because he's 2, and that's a great thing to do. Picks up microbes, sticks his finger right in his nose, he's just put it in a portal of entry. Pick up microbes on our hands. We don't wash our hands before we eat. We've just put those microbes on our food. We've just put it into a portal of entry. So keeping our microbes on our skin and not in a portal of entry is a really good idea. All right, so the dermis, our skin layer, we've got our dermis and then our epidermis, okay? And we'll look at a lovely picture of that. On our epithelial cells, we've got keratin, and that is a very protective protein. The shedding of these skin cells helps prevent microbial growth, and the dryness of the skin helps prevent certain microbes from growing. So here's our epidermis. Okay, that's that thick layer. It's got keratin, and then below that is the dermis. So this is a nice thick layer preventing the invasion of microbes unless it is broken in any sort of way. Within portals of entry, so you can think about all your portals of entry we talked about, right? We've got a couple of big portals of entry. All of those portals of entry are usually lined with what we consider mucous membranes. So what are mucous membranes? Think of the inside of your mouth. Think of that inside of your nose, that kind of warm, squishy kind of cells that have a little bit of a layer of mucus on them. Inside our eye, so our eyes are another portal of entry for some microbes. So if we touch the surface and then rubbed our eyes, that could be a portal of entry. So your digestive tract, your urinary tract, your eyes, those mucous membranes, they're there to help protect microbes from coming farther into your body and causing infection. So they prevent attachment to those surfaces because it's kind of a slick layer that the microbes can attach to. Mucus itself is made out of glycoproteins. It helps trap microbes and prevents those kind of mucous membranes from drying out. The lacrimal apparatus that helps with the tears in your eyes to start washing out any sort of microbes that potentially can get into your eye. So let's talk about snot and nose hairs. Okay. Think about your nose hairs. So your respiratory tract, your nose hairs are preventing any sort of large particles, hopefully from getting into your respiratory tract through your nose. So those hairs are able to trap larger particles. They mix with the mucus inside your nose, and they make kind of a lovely boogery substance that helps prevent it from getting any farther inside your respiratory tract. The flow of mucus and fluids from your nose, anytime you have some sort of antigen coming into your body, you can think about this for any of you guys who have allergies, is called rhinitis, and that's the flowing of the mucus and any sort of fluids from your nose, and that helps flush out that general area. So if you went outside and you're allergic to pollen, like I am, take a nice deep breath in. We've got all that pollen that comes in. You start getting a runny nose. That's your immune system trying to help you flush out anything from getting farther into your body. Your respiratory tract has epithelial cells that have cilia on them, so ciliated epithelial cells. They help move any sort of particles that have been trapped lower in your pharynx, so they've come a little bit farther down, and they're trying to move them up, preventing them from getting into your lungs. any sort of irritation in your throat or in your nasal passages that will induce sneezing or coughing to help project out those particles so that they don't come farther into your respiratory tract. So all of that is trying to help prevent anything from getting farther into your respiratory tract and causing disease. Here's a picture of that ciliated epithelial cells there. It's this is kind of a cartoon drawing of it, helping push out any sort of particles that may have gotten down into your respiratory, down farther into your trachea, your larynx down there. Other physical factors, your earwax, vaginal secretions, even the process of vomiting is for your body to try to get anything out that is deemed potentially harmful out of your body. So if you've eaten something that potentially has a toxin in it, you're going to try to eliminate it as fast from your body as possible and get it out of there. So those are the physical barriers. Then we're going to move into chemical barriers. So these are chemicals that are inside your body that help prevent the growth of microbes. In your tears and in your saliva, we have lysozyme and defensins. So lysozyme, remember how we looked at that in lab so very long ago, are able to break down peptidoglycan in bacteria. Defensins can also lyse bacteria and fungal cells. So those are just non-specific chemicals. They don't care what kind of bacterial fungal cell is. It's going to try to just eliminate any of those microbes from that area. Having the high pH in your stomach helps get rid of some microbes in the food that you eat. Bile also helps that. The acidic pH in the vagina also helps from growth of any sort of foreign microbe. Okay, so all those guys, we're not going to talk about the genetic barriers, but this is, I actually just read an article yesterday, I can't even remember, all the days kind of run together, that there is a group now looking at the genetics of people who've been infected with coronavirus and looking to see if they can see any sort of genetic patterns with people who got severely sick versus people who don't. There's already lots of consortiums out there. If you wanted to have your DNA as part of a database that have large databases of different people's genetic backgrounds, and now they're trying to cross-reference those with people who have been infected with coronavirus to look and see if they can find any sort of patterns that lead to more severe versus less severe infection. So they're really looking at those genetic factors and how they're involved in this infection. So that's, when we're talking about genetic factors, that's what we're kind of talking about. The resistance to HIV. So some people have the deletion in CCR5, which is one of the receptors that HIV uses to gain entry in cells. They were naturally born with that allele of that receptor, and they are HIV resistant. So some people will have a certain genetic mutation that makes them less susceptible to certain diseases. So that's what we're talking about, genetic barriers. Okay. So first line of defense, physical barriers, chemical barriers, and then those genetic barriers. Then if one of those is broken free, so let's think about our skin. We get a cut on our skin. We've now eliminated our skin as a defense mechanism. What happens next? How do we prevent microbes from getting further into our body? That's when our second line of defense comes in there. And that includes our inflammatory response, something called complements, our cells called phagocytes, and then interferons that are important in viral infections. So let's first talk about our immune cells. So most of you guys that have taken physiology before, and all of you guys in immunology right now, have probably come across the names of most of the cells involved in the immune system. So we're going to go over this kind of generally. The function of your immune system is to do surveillance, right? At all times, there should be immune cells going through your body looking for potential invaders, whether they're from breaks in the skin, whether you have ingested them or inhaled them. Your body is going to be constantly looking for any sort of foreign invader. they're going to check and see, is this foreign or should this be here? Is it self? Is it something that's good? And then if anything is deemed foreign and harmful, let's seek out and destroy that foreign invader. The cells of your immune system, we often call them white blood cells. That's kind of an archaic term. When scientists a long, long time ago looked into blood, they saw your erythrocytes, your red blood cells, and then they saw all these other ones they termed white blood cells. This kind of more scientific name for them is leukocytes, and those are the cells involved in your immune system. Certain members are always circulating throughout your body, checking for these foreign invaders, and they're very important in that they need to be able to distinguish between what is self, so what should be in your body, and what is foreign. And if they have a breakdown in this, this could potentially lead to what we call autoimmune disease, where your immune system is thinking that something that's supposed to be in your body is actually something harmful and foreign and needs to be destroyed. So how do they tell the difference? We'll go more into that a little bit more in a little bit, but let's talk about your blood cells. The blood cells that are part of your immune system and your red blood cells and platelets all come from a hematopoietic stem cell. So this is something found in the bone marrow. And it gives rise to two different types of stem cell, the myeloid stem cell. The myeloid stem cell ultimately makes red blood cells, platelets, and some of your innate immunity cells. And then the lymphoid stem cell, which ultimately becomes your B cells, your T cells, and your natural killer cells. And those are all involved in your specific immune system. This is just a lovely picture from another book, kind of gives you a cartoon drawing. Here's our pluripotent stem cell, meaning it can be made into lots of different types of cells. It starts down the road of the myeloid stem cell, then here are the choices that it could be made into. If it goes down the lymphoid stem cell, here are the choices that it could be made into. Okay. So we'll start talking about mast cells, eosinophils, basophils, all these lovely cells, macrophages, T cells, B cells, and NK cells. So all these play a role in the immune system. So your erythrocytes or your red blood cells and platelets aren't necessarily part of your immune system. Your erythrocytes, your blood cells delivering oxygen, taking out CO2, your platelets help in blood clotting. So we're going to focus on the rest of these guys here. Mast cells, for those of you guys that are in immunology, heard all about mast cells this week. I hate mast cells. No, I'm just kidding. Mast cells are the ones responsible for allergic reactions. So any of you who suffer from allergic rhinitis like I do can thank your mast cells for reacting to all the pollen. What did Dr. Thompson say? The trees that are undergoing reproduction right in front of our eyes and shooting out all this pollen, making us all upset. Anyway, the pollen from those trees are causing those mast cells to release their granules and cause inflammation, also known as allergies. Granulocytes, we call them granulocytes because under the microscope we can see these granules. They include neutrophils, basophils, and eosinophils. Neutrophils are phagocytes. We'll talk about what phagocytes do in a little bit. Basophils are important in inflammation. They also have a role in allergic reactions, as well as eosinophils. Eosinophils we think are important in worm and fungal infections, trying to fight those different types of infections off. Macrophages are very important phagocytes. They are also what we call antigen presenting cells, and that's going to be important when we're talking about specific immunity. They help in the process of turning on your specific immunity. So they're really important for not only doing phagocytosis, engulfing microbes and killing them, but turning on the specific end of your immune system. Dendritic cells are related to macrophages. They're found in our tissues, and they're also important in antigen presentation. Then your specific immunity Those include your B and T cells, your B and T lymphocytes, and your natural killer cells. When we get it to a specific immunity, we'll talk all about what those guys do. T cells are mature in the thymus, hence the T cell. The B cells mature in the bone marrow, and the B cells' big job is to produce the antibodies for our specific immunity. Lovely pictures in case you ever wanted to know what they look like underneath the microscope. The basophils, see those are the granules in the basophils, eosinophils, neutrophils, or basophils, eosinophils, you see the granules there. Here's your macrophages, your dendritic cells, T cells, B cells, and NK cells. How do we get our immune cells to where they need to go? Well, they've got a very specific highway that they like to travel in. They go in the endoreticular system, so kind of thinking in between the cells and the tissues. They go in our bloodstream, they go the lymphatic system, and then all of that extracellular fluid. If you could imagine getting a cut on your finger and it starts to swell, we've got the increase of fluid to that general area, and that brings a lot of our immune cells to that area. So our immune cells are constantly traveling, doing their surveillance, looking for invading microbes, and then if an invasion is found, drawing other white blood cells to that area of invasion, hopefully killing any sort of microbe before it gets further into the body. The lymphatic system, please don't memorize all the terms on here. It's just to give you a general idea of your lymphatic system. Lymphatic system is bringing the flow of lymph, lymphatic fluid in between your lymphatic system, where it's going to circulate between all your immune organs and then your bloodstream. So bringing fluid in from your lymphatic system, your bloodstream, draining off any sort of fluid into your lymphatic system. So what is lymphatic system doing? Helps bring some fluid back into the circulatory system. So imagine if you're getting very swollen, we got to get rid of that fluid somehow. Your lymphatic system helps you do that. Lymphatic system is going to help do that surveillance. So all those lymphocytes are going to be traveling throughout your body, looking for invaders. If they find invaders, they're going to bring them back to those lymphatic organs like the spleen and turn on the rest of your immune system. So what is lymph? Lymph is this like fluid stuff, got some salts, got some proteins, and got a whole lot of white blood cells. You can imagine when you're going to the doctor, they often fill what we consider lymph nodes, which are the collection of your lymphatic system where microbes are brought. And if they're swollen, that's one of the signs of infection. Unlike your circulatory system, lymphatic system only flows in one direction, out to in. And it does not pump when your heart beats like your circulatory system. So the only way your lymphatic system circulates is if you move. So you get up and move. So if you're stationary for any period of time, you don't get drainage of your lymphatic system. You don't get the movement of that lymphatic fluid, and you'll often get swollen. Okay, so the primary organs of the lymphatic system include your thymus and your bone marrow. And then our secondary organs, lymph nodes, our spleen. And then we've got all this lymphoid tissue associated with different parts of our body. And you can think of this as like, these are the portions of your body where you potentially encounter harmful microbes, right? The skin, so we call that skin-associated lymphoid tissue or SALT. Your gut inside your intestinal tract, right? That's where potentially harmful microbes could be. We call those gut-associated lymphoid tissues. And there's these little tiny collection of lymphoid tissues called the Peyer's patches. And some enteric microbes like to invade those Peyer's patches and live there. So they're actually living inside those lymphoid tissues inside your intestine. And then your MALT or your mucosal associated lymphoid tissue. Again, those mucosal membranes, potential interaction with harmful microbes. We've got lymphoid tissue that is associated with those interactions. So the lymphatic organs, their job is to survey seek out and destroy microbes, and then send out any other white blood cells if there is a pending infection. Example of the thymus gland. Here are the lymph nodes. So we've got lymph nodes in our armpits, we've got them in our neck. Tonsils, Peyer's patches, appendix, all lymphoid organs. Spleen is a very important portion for filtering our blood. One of its big jobs is to get rid of old red blood cells. So any sort of red blood cell that needs to be destroyed, it's gotten old, will filter through the spleen. There's also immune cells that have phagocytized bacteria or other pathogens have brought them to the spleen to activate some of the other immune cells. Malt and galt, again, mucosal associated. Malt includes those tonsils in there. Hopefully none of you have gotten your tonsils out. You can survive without your tonsils, but they are important in your immune system, at least a little bit for processing antigen that or microbes that have come in through your mucosal tract. Galt, appendix, Peyer's patches, all part of those gut-associated lymphoid tissues. Okay, so I think with that, I know we still have a little bit of time left. I'm going to go ahead and stop there before we go into inflammation. We'll pick up inflammation on Tuesday. I might not get through all of these slides before the exam. That's okay. We'll go ahead and stop wherever I get through, and we'll pick up some of the stuff after the exam when we're talking about specific microbes. Does anybody have any questions up until this point? No questions? Okay. So look for this afternoon videos from lab. Those will populate either on Moodle or in your folder, depending on what size they are. And then pictures of some of the tests will pop up today for exercise 34, 35, and 36. I'm going to go talk to Raina right now to see if the microscope did come or if it was something like a book and boring that came in the mail today. So I will repost those gram stain pictures as soon as we get that microscope in. It may be tomorrow, it may not be today, but we'll see. Fingers crossed it came in today. All right. Send me an email if you have any questions. Oh, got a chat on here. Hold on one second. Yes, for your lab report, you're going to pretend like you did everything yourself. Ta-da! So hopefully you can get from my videos, all of the protocols should be in the lab manual, but hopefully you can get from the videos how I did each one of the inoculations. Most of the materials and method is going to be you inoculated this tube and grew it at this temperature. It's not going to be very detailed. Some of the additional tests that I'll post today, like the methyl red and the Vogue's posture, you have to add some stuff to the tube to complete the test. So those will have a little bit more directions in the materials and methods, but you guys are going to pretend like you did them yourself. Just like a cooking show, you're going to watch me do the video and take credit for it. It's okay. Any other questions? No. So there's no other assignment you need to submit except the lab report for the unknown. That's all you need to submit for that. The chart from the book, you'll include that in your lab manual, but you don't need to do any of the questions or anything from the lab manual. It will just be the lab report. Yeah. Other questions? All right, if you think of anything else, just email me. Hope you all are doing well. Oh, yes, so your lab report will include pictures. They will be my pictures. You can give me credit for them. No, so the pictures that I put in the folders, you guys can use those for your lab report. That's what I expect you to do. Some of them are a little bit big, so if you want to use your cropping skills, you're welcome to do that, make them a little bit prettier. You'll need to put them in figure form with a like a title, like figure 1, and then a description of what's in there. The unknown lab report has like an example of that on there, so you guys can copy that example for each one of those figures. Yes, so you guys will pretend like those are your results and you'll put them in your results section. Any other questions? Nope. All right, you guys enjoy the rest of your afternoon. I will see you later. Bye-bye.